This video presentation covers the basic early events of embryogenesis from fertilization to uterine implantation and to formation of 3 embryonic germ layers which give rise to the tissues and organs of the body. It concludes with a summary of body folding that converts the flat embryonic disc into a tubular body form. This will set you up for future discussions of organ systems development. In the background of these descriptive events is a richly complex orchestration of changes in cell shape, genetic signaling, secreted growth factors, programmed cell death, and a multitude of inductive events. A focus on these details is outside the scope of this presentation. Most contemporary embryology textbooks contain such information, and you are encouraged to explore these if you are interested. This list of learning objectives will guide your learning of the content of this presentation. Fertilization occurs in the ampullary region of the uterine or fallopian tube when a sperm penetrates the zona pellucida of an oocyte and their cell membranes fuse. This union reestablishes the diploid complement of chromosomes 46 and produces a single-cell zygote. Within 24 hours, the zygote divides or is cleaved into a 2-cell structure, and repeated mitotic divisions produce increased numbers of cells within the zona pellucida. Each daughter cell formed is referred to as a blastomere. While these divisions are occurring, the conceptus is transported toward the uterine cavity via ciliary action of the cells lining the uterine tube. By 4 days post-fertilization, the embryo consists of 16 to 32 blastomeres and is called a morula, Latin for mulberry. The morula enters the uterus between day 3 and 4 after fertilization. At this time, the morula develops a fluid-filled cavity, the blastocyst cavity, and is called a blastocyst. Blastomeres subsequently organize into an inner cell mass called the embryoblast, and an outer cell mass called the trophoblast. The trophoblast becomes the future fetal portion of the placenta. Embryoblast cells are pluripotent, meaning they are able to differentiate into ectoderm, mesoderm, or endoderm. By day 5, the blastocyst hatches out from the zona pellucida through enzymatic action and is now free to attach an implant into the endometrial lining of the uterus. The blastocyst typically implants into the posterior superior wall of the uterus by day 7. It is not uncommon for the conceptus to embed into tissue other than the uterus should there be some interruption of blastocyst transport. Such extrauterine pregnancies are referred to as ectopic pregnancies. The majority of ectopic pregnancies occur in the uterine or fallopian tube and are called tubal pregnancies. Other possible sites include the surface of the ovary or peritoneal lining of the abdominal cavity. The most common clinical presentation of ectopic pregnancy is first trimester vaginal bleeding and/or abdominal pain mimicking appendicitis. Early stages of ectopic pregnancy, however, may also be asymptomatic. The diagnosis of an ectopic pregnancy is based upon a combination of transvaginal ultrasound findings and serum levels of human chorionic gonadotropin. An ectopic pregnancy may be unruptured or ruptured at the time of presentation to medical care. Tubal rupture or rupture of other structures in which an ectopic pregnancy is implanted can result in life-threatening hemorrhage. Let us return now to more completely describe the blastocyst, the process of implantation, which begins at the end of week 1, and growth of the embryo during week 2. For orientation purposes, again, we have our blastocyst that is beginning to embed into the endometrial lining of the uterine cavity, and that lining covers our uterine wall. By the time the blastocyst arrives in the uterine cavity at the end of the first week, the functional layer of the endometrium is prepared to support blastocyst implantation. The outer cell mass, called the trophoblast, proliferates and differentiates into the syncytial trophoblast, the light green cells in the diagrams, and the cytotrophoblast, indicated by the slightly darker green cells. The syncytiotrophoblasts is derived from the cytotrophoblasts and represents a multinucleated syncytium that continuously invades and insinuates itself into the wall of the uterus and erodes endometrial blood vessels and glands. This erosion process produces spaces or lacunae within the syncytiotrophoblast, which become filled with maternal blood and glandular secretions. These spaces eventually coalesce into lacunar networks that will become the uteroplacental circulation system. By day 9, the syncytiotrophoblast entirely encloses the blastocyst, and the embryo becomes fully implanted in the endometrium. The site of implantation is temporarily marked by a coagulation plug at the endometrial surface. Early in the second week, the embryoblast differentiates into two distinct epithelial layers: the epiblast, indicated by the blue cells, and the hypoblast, or primitive endoderm, indicated by the yellow cells. Together, these two layers form a structure called the bilaminar embryonic disc. With its formation, the dorsal-ventral axis of the embryo is established. That is, the epiblast is dorsal and the hypoblast is ventral. By day 8, a new fluid-filled cavity forms between the epiblast and the overlying cytotrophoblast, called the amniotic cavity. By the 8th week, this cavity will entirely enclose the embryo. Proliferating cells from the hypoblast layer migrate outward in an initial wave internal to the cytotrophoblast to line the blastocyst cavity. Let's focus now on events during the remainder of the second week on the next few slides. By days 10 or 11, migrating hypoblast cells complete their enclosure of a space within the blastocyst called the primary yolk sac or primary umbilical vesicle. This layer of lining cells is referred to as Heuser's membrane or the exosomic membrane. At the same time, mesoderm outside this membrane, known as extraembryonic mesoderm, fills the remainder of the blastocyst cavity between Heuser's membrane and the cytotrophoblast. This extraembryonic mesoderm represents a loose network of cells and is derived from the hypoblast. Spaces subsequently appear within the extraembryonic mesoderm and coalesce to form a single fluid-filled cavity called the chorionic cavity. Two layers of extraembryonic mesoderm can be described, one layer that surrounds the primary yolk sac and another that lines the inside of the cytotrophoblast. A second wave of migrating hypoblast cells line a smaller space called the secondary or definitive yolk sac. In this process, the primary yolk sac becomes pinched off and degenerates into a collection of vesicles. As we will discuss in future presentations, the definitive yolk sac is a major site of early hematopoiesis in the embryo and the source of primordial germ cells for the gonads. By the end of the second week, the conceptus consists of a two-layered embryonic disc separated by two cavities, a dorsal amniotic cavity and a ventral yolk sac or umbilical vesicle that is suspended in the chorionic cavity by a thick stalk of extraembryonic mesoderm called the connecting stalk. As you've probably discerned, week 2 is a busy week with simultaneous activities occurring in the trophoblast and the embryoblast. Fortunately, much of the descriptive events can be boiled down into a rule of twos to help with the management of the details. During this week, the embryoblast gives rise to two layers, the epiblast and the hypoblast. The trophoblast forms two layers, the cytotrophoblast and the syncytiotrophoblast, which plays a key role in eroding into the endometrial lining. Two yolk sacs or umbilical vesicles are formed. An initial primary yolk sac, which becomes pinched off to form a secondary, smaller, or definitive yolk sac. Finally, two cavities are formed, the amniotic cavity and the chorionic cavity. We now turn our attention to a further discussion of the development of the uteroplacental circulation. Simple diffusion serves the nutrient and growth needs of the embryo during the first week of development. Thereafter, the embryo requires a more efficient method to promote further growth. This involves the establishment of the uteroplacental circulation. The essential details of this process are shown on the next couple slides. Recall from a previous slide that trophoblastic spaces or lacunae formed within the syncytiotrophoblast join and merge with maternal blood vessels. Between days 11 and 13, the cytotrophoblastic cells proliferate and form finger-like projections that grow into the overlying syncytiotrophoblast and extend into the trophoblastic lacunae to form primary chorionic stem villi. By day 16, extraembryonic mesoderm proliferates and penetrates the central region or core of the primary villi to form secondary chorionic stem villi. By the end of the third week, blood vessels arising within the extraembryonic mesoderm connect with vessels forming in the embryo proper and transform the secondary villi into tertiary chorionic stem villi. The primitive heart begins to beat on day 22 to commence a functional uteroplacental circulation system. Gases, nutrients, and waste products from fetal blood vessels exchange with gases and nutrients from maternal blood contained within the trophoblastic lacunae, and these diffuse back and forth across the layers of the tertiary villi. We move now on to events in week 3 of development. Gastrulation is the first major developmental event in the early part of week 3. In this process, the bilaminar embryonic disc is transformed to a trilaminar embryonic disc consisting of 3 primary germ layers, ectoderm, mesoderm, and endoderm, from which all the tissues and organs of the body form. The next set of slides describes the essential details of this process. We should add here the fact that beginning in weeks 3 through 8, a period known as the embryonic period, all the major organ systems begin to develop. It is also a period of vulnerability or risk to the embryo should it be exposed to teratogens or agents that may disrupt organ development. This week also coincides with the first missed menstrual period when the woman may first become aware she may be pregnant. At the end of week 8, the embryo has a distinct human appearance. This is an expanded dorsal view of the developing embryo as the bilaminar disc. Around day 15, a midline thickening appears on the dorsal surface of the embryonic disc due to proliferation of epiblast cells called the primitive streak. This marks the beginning of gastrulation. The primitive streak contains a groove, the primitive groove, which ends at the primitive pit, a depression within the primitive node. The primitive node is an expansion of the primitive streak at the head or cranial end of the streak. The primitive streak is important in establishing the axes of the developing organism, left-right axes, cranial-caudal, dorsal-ventral, and medial-lateral axes. In this slide, we describe the formation of the 3 germ layers. The images here show cross-sectional views of the embryonic disc transforming from bilaminar to trilaminar. The 3 germ layers are formed by epiblast cells. On days 14 and 15, there is a first wave of epiblast cells, in illustration A here, that migrate toward the primitive streak and into the primitive groove and displace and replace hypoblast cells to form the definitive endoderm. On day 16, a second wave of epiblast cells migrating through the primitive streak enter and fill the space between the epiblast and the definitive endoderm to form the intraembryonic mesoderm. Once formation of the endoderm and mesoderm layers is complete, epiblast cells cease to move toward the primitive streak, and the remaining epiblast becomes the surface ectoderm. This slide lists several of the tissue and organ system derivatives associated with each germ layer. We will refer to this information numerous times as we progress through organ development in later presentations. It is important to realize, however, that few structures fully arise from a single germ layer. Rather, two or more germ layers work in unison to form organ rudiments. For example, the epithelial lining of the digestive system is derived from endoderm, but the smooth muscle walls of the gastrointestinal tract arise from mesoderm. The primitive streak is active during the remainder of the third week in forming mesoderm that migrates in all directions beneath the epiblast layer, except at the oropharyngeal and cloacal membranes at the cranial and caudal portions of the embryo, respectively. Thereafter, the primitive streak decreases in size, degenerates, and disappears by the end of the fourth week. Occasionally, this degenerative process is incomplete, and remnants of the primitive streak persist to form a sacrococcygeal teratoma that appears as a mass extending from the caudal end of the fetus. These are the most common congenital tumors in neonates and represent a type of germ cell tumor. They occur in about 1 in 35,000 to 40,000 births and have a female predominance. Most sacrococcygeal teratomas are benign, but may become malignant with increasing age and therefore are usually removed shortly after birth. These tumors are generally identified in utero by prenatal ultrasound. Because the primitive streak cells are pluripotent, contents of these tumors can include fully functional glandular structures such as pancreatic Langerhans cells, sebaceous glands, bone, teeth, or hair. This slide summarizes the development of an important structure called the notochord. Some of the ingressing primitive streak cells migrate cranially from the primitive node and give rise to a midline mesodermal cord of cells called the notochordal process. This transforms into a solid cylinder of cells called a notochord. The notochord elongates between the ectoderm and endoderm layers until it reaches the prechordal plate. The prechordal plate is a thin circular area of fusion of ectoderm and endoderm with no intervening mesoderm. This plate becomes the oropharyngeal membrane which separates the primitive mouth from the primitive pharynx. During the fourth week, it ruptures to provide continuity of those structures. Caudal to the primitive streak, there is another circular area called the cloacal membrane. Again, this is an area of fusion of ectoderm and endoderm with no intervening mesoderm. This is the future site of the anus. We'll refer to these areas again in future presentations covering the development of the face and development of the intestinal tract. The notochord has several important functions. First, it serves to define the longitudinal axis of the embryo, and secondly, it is a primary inducer of the development of the central nervous system, and thirdly, it plays an important role in the formation of the vertebral column and its associated musculature. Most of the notochord degenerates as the vertebral bodies form. However, portions remain that become the nucleus pulposus, or the jelly, of intervertebral discs. This slide summarizes the key events in week 3. The bilaminar embryonic disc becomes converted to a trilaminar embryonic disc with the appearance of the primitive streak and migration of epiblast cells to form the 3 germ layers. At the end of week 3, the embryo exists as a flat oval structure. This quickly changes in week 4 to a tubular body plan, as we will see shortly. The notochord is formed as a midline rod of cells, which induces the overlying surface ectoderm to thicken as the neural plate, the precursor of the central nervous system. This occurs in the process of neurulation, an event you will learn about in the movement and mechanics module. The notochord also plays a role in organizing the development of the vertebral column and its associated musculature. Finally, the intraembryonic mesoderm formed earlier in this week begins to differentiate and assume different names, with each subdivision having different fates and functions. These will be discussed on the next slide. Recall in a previous slide that with the exception of the oropharyngeal and cloacal membranes, intraembryonic mesoderm migrates in all directions beneath the ectoderm and endoderm layers. This mesoderm subsequently differentiates into paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm, with each subdivision giving rise to unique structures. The paraxial mesoderm, organized immediately on each side of the notochord, gives rise to the mesoderm of the head, to somites, and limb musculature. Details of this type of mesoderm will be given in the Movement and Mechanics module. Just lateral to the paraxial mesoderm is a longitudinal ridge or column of mesoderm called intermediate mesoderm. This produces the urinary system and parts of the genital system and will be discussed further in the Sexuality and Reproduction module. Most laterally is the lateral plate mesoderm. This is a flattened sheet of mesoderm that later splits into two layers, somatic mesoderm and splanchnic mesoderm. These layers will be discussed in their appropriate modules. In this last slide, I simply want to develop for you the notion of conversion of a flattened structure into a tubular structure by virtue of body folding processes. As we learn about the development of different organ systems, we will further elaborate on how such foldings move structures toward their adult positions and relationships. Recall that the trilaminar embryonic disc was a flat oval structure at the end of week 3. Rapid growth of the dorsal embryonic disc and amnion during week 4, relative to the yolk sac, causes the head end, tail end, and lateral edges of the disc to fold or bend in a ventral direction. These edges from opposite sides will eventually fuse in the ventral midline, except at the site of the future umbilicus. This creates a tubular body form with an outer tube representing the external surface of the embryo, enclosing an inner tube represented by the gut tube or primitive gastrointestinal tract. Craniocaudal folding occurs along the mid-sagittal plane, best seen in figures A, B, and C. Lateral folding occurs also in a ventral direction and brings the edges of the embryo toward each other in the midline, best seen in the cross-sectional views in figures D and E. Gaining a basic appreciation for these types of folding is sufficient for our needs now. This ends this video presentation covering early embryogenesis. I wish you well with your studies.